alum, and he was here when I first started. And he was finishing up his thesis at that point, but he really helped kind of welcome me to Moss Landing, so I have a very soft spot in my heart for Keith. Um, I'm sure you guys know when you show up somewhere new and you don't know what's going on, he helped me figure out how to set up my computer, all that kind of stuff. He helped me teach the first time, or he was my TA the first time I taught marine mammals and birds, and so he helped me find specimens and figure out what kind of labs were possible. And he also was running the stranding program, which I had inherited from Jim. And he, before he left, trained up one of my new students so they can continue to run that program. And our lab has continued to run that program to this date. Since leaving here, well, his thesis while he was working here was looking at sex-specific diet in California sea lions using molecular techniques. And he graduated in 2016, and then he went on to Louisiana State University, where he continued to study foraging ecology and diet. But he went and worked with Mike Polito, who is an expert in stable isotope analysis, and developed a new tool kind of to put in his toolbox. So he studied the diet of gray seals using stable isotopes, and I think you continue to use DNA if I'm correct. So kind of used his previous and got a new skill. He graduated in 2000 and returned back to Santa Cruz. 2020. Sorry, 2000. You're old. There's a 20 in there. He graduated in 2020 and came back to Santa Cruz, where he has been working as a postdoc with data from a species that I'm very attached to, the elephant seal. So he's been continuing to do some of his DNA analysis, looking at the bottleneck, and he's also trying to look at what are some of the traits that make some elephant seals so successful. And now he's in June moving up to Oregon to start a new postdoc, working with epigenetics of aging in whales and dolphins. See, I just asked him that, so that's a new addition. I'm not as familiar with that. But with that, I will introduce Keith.
Wow. Thank you so much, Geeta, for that warm introduction. It's great to be back at Moss Landing Marine Labs. So I'm excited to share with you one of my dissertation chapters looking at foraging ecology in gray seals. So to start, top predators are a critical component of marine ecosystems. I think we're all pretty familiar with the idea that they exert top-down level forcing on lower trophic levels through consumption and also competition. We also know that top predators act as sentinels of ecosystem health. So by studying them, we get an understanding about what the ecosystem is doing and kind of how things are going, especially in the context of continuing environmental change as well as human disturbances. And we know that they can interact with humans in a variety of manners. People can see them through tourist activities, and one of the other things that we know that they do is that recovering marine mammal populations are increasingly interacting with fisheries. Many species of marine mammals were reduced through exploitation, through either direct harvest or competition. Since protections have been enacted in the US and across the globe, these recovering populations are increasingly interacting with fisheries both directly and indirectly. So directly, they're consuming prey and they can also damage gear. Indirectly, they could accidentally be caught in the gear, and sometimes they could be intentionally killed, which kind of sets up an interesting conundrum, because how do you balance managing a protected species but also a commercially important species for fisheries? So it brings to mind that better management is necessary for both marine mammals and the prey that they consume. So focusing on gray seals, they're ubiquitous across the North Atlantic. They're found from now the mid-Atlantic region in the US, across the North Atlantic into the Baltic Sea, and there are 3 genetically distinct populations. In New England, they were hunted until near extirpation, until about the 1950s, by a state-sponsored bounty program, so you can cut off a nose or a flipper of a seal and get a small bounty for it. But with the enactment of the Marine Mammal Protection Act, they were protected, so those bounty programs ended. And as a result of this protection at the national level, as well as immigration from the rookeries off of the east coast of Canada, the gray seals in the US have kind of repopulated and rebounded in a stunning conservation success story, but it has prompted kind of renewed concerns about the presence of this predator in their historical ecosystem. And a lot of these historical concerns have focused on the interaction between gray seals and fisheries, particularly for ground fishes like Atlantic cod, which you may be familiar with as being very commercially important, as Cape Cod got its name from the cod fishery there. But that's not the only issue between seals and humans. So in addition to kind of this historical and continuing competition between seals and fisheries, There's also some more recent issues that are coming up as humans and seals come into continuing and increasing contact. One of these is the idea that seals may be polluting the local ecosystem by pooping into the water. Their also continued presence on tourist beaches kind of sets up interesting conflicts between people, their dogs, and seals when they're all just trying to rest on the same beaches. And kind of most recently, there's the increased occurrence of large sharks off the coast of Cape Cod, which is again setting up kind of interesting issues about human safety versus kind of the recovery of an ecosystem that's been historically over-harvested and impacted by human activities. So focusing again on this issue about seals and their diets, what we do know kind of historically from the New England area as well as across their range is that gray seals are considered benthic generalists. So they're eating a wide variety of prey typically associated with the bottom of the seafloor. Across the range, they're eating typically sand lance or related species, flatfishes like flounders, cods, hakes, as well as skate tends to be an important prey item across their range. But a lot of these studies tend to use just one technique, which is to identify prey remains from a scat or a stomach sample of a wild or a bicaught individual. But that's not the only way that you can study diet in a marine mammal. You could try to visually observe predation events at the surface, but this doesn't really work for seals since they tend to catch their prey under the water column and consume their prey at depth. As I mentioned, you can analyze prey remains. So here in the image, you can see fish otoliths or ear bones and squid beaks, as well as some other, I think this is a denticle from a skate here in the center, and there's other random hard parts. I think this is a mussel shell that showed up in one of our samples. And this is great because you can quantify and estimate the biomass of prey that were consumed, but it does reflect just a very short time frame of prey consumption, typically on the order of a couple days. So that doesn't really tell you about longer-term trends in diet on the order of weeks to months or even years. So, and also hard parts tend to overestimate prey remains that tend to get stuck in the digestive tract. So a good example are squid beaks. They can get stuck in the digestive tract and may kind of overestimate the importance of that prey item relative to what's going on a different time scale. So an alternative method is to use DNA metabarcoding. So you amplify small segments of DNA that are diagnostic to ideally the species level out of a community diet sample. And so this is great because you improve the taxonomic resolution of the prey that you get from your diet sample. But similar to the hard parts method, it does reflect just a short time frame of diet, typically on the order of about 3 days. And kind of the major drawback to DNA metabarcoding at the moment is that it's still not directly quantifiable. So you can't exactly take the proportion of sequence reads that you get and relate that to a number or biomass of prey that are consumed. So an alternative method is to use stable isotope analysis. This does get around some of the issues that I've mentioned so far. So you may be familiar with stable isotope analysis, and it's based on this principle that you are what you eat, plus a little difference due to kind of body chemistry reactions that go on. So the two most typically used isotopes for these kind of studies are carbon-13 and nitrogen-15. Carbon stable isotopes give you an indication of the primary production source in the ecosystem where your predator has been foraging. So in the little schematic here, you can tell apart phytoplankton from macroalgae as the base of the food web that your predator is foraging in. And nitrogen stable isotopes give you an indication of the trophic level of the predator relative to the prey that it's consuming. And the time that's reflected by stable isotope analysis depends on the tissue turnover rate. So at the cellular level, the cells tend to replace themselves within the tissue, and this gives you a longer-term trend of what the predator was consuming. And this could be anywhere from a couple days to weeks for certain tissue types to longer, potentially years over the lifetime of an animal, depending on how the tissue is replaced and how long it's growing. So stable isotope analysis is really useful for getting at longer-term trends in diet. And additionally, while stable isotopes can't tell you directly what prey were consumed, there are analytical methods to estimate this, and this is through stable isotope mixing models. So you can take stable isotope values for your predator as well as the prey that it's potentially consuming and use mathematics to estimate potential diet contributions for the specific tissue you had. And recent advancements with Bayesian frameworks have now allow you to add uncertainty estimates. So you don't just get a point estimate about what the potential contribution is, you now have an idea of the certainty around that estimate. So that's pretty useful compared to kind of the first iterations of these models. Additionally, there's isotopic niche analysis, so you can take the stable isotope values and use those to kind of estimate different aspects of the ecology of the animals that you're working with. I won't be presenting this today, but I did use this with other parts of my dissertation research for gray seals. So I've talked about a couple different methods that you can use to analyze the diet of marine mammals or any predators. And while it's often suggested, it's still not commonplace to integrate multiple diet methods together. When it is done, it tends to be hard parts paired with stable isotope analysis, or recently with DNA metabarcoding, as it's becoming a little bit easier to implement and understand. It's rarer, but stable isotope analysis and DNA metabarcoding are sometimes paired together. So in this figure here from this paper, you have the isotopic proportion of C4 plants and the DNA sequence abundance for this group, which I think are grasses, for different herbivores on the savanna. So you can see there's kind of a great concordance between the proportion of C4 plants and the reeds associated with grasses that are obtained from the same sample type. And there's a few studies, but an increasing number, they're integrating all three methods together. So you can overcome kind of the short time frame bias, you can improve the taxonomic diversity of the prey that are being consumed, and just have a more holistic understanding about what your predator is consuming. So again, stepping back to gray seals in the US, most of the previous studies, and there are not that many, have generally used a single method to identify diet items. So kind of the first study just used hard parts. What I would consider the foundational study that a lot of us have recently based our work off of also just used hard parts from scats and stomachs of bicaught individuals. And there's been some preliminary work with fatty acids also from bicaught individuals. Recently, we've been applying stable isotope analysis with mixing models to estimate prey contributions for mothers when they're pregnant. And then most recently, there have been taxon-specific efforts to identify prey with a DNA metabarcoding framework. So as part of my dissertation research, I wanted to integrate information from all three techniques to kind of get the most comprehensive understanding of gray seal diet off of Cape Cod to date. Because we were integrating information from all these methods together, we also examined the efficacy of including information from hard parts and DNA into informing the stable isotope mixing model, which I'll explain a little bit more later. And finally, we wanted to use this information not just to say what those seals were eating, but also to investigate this idea about overlap between marine mammal consumption and fisheries consumption in the same waters. So we used information from Massachusetts commercial fisheries. So we collected seal scats off the coast of Cape Cod, Massachusetts, between January 2016 and March 2017. So we saved a portion of the seal scat for stable isotope and DNA metabarcoding analyses. The rest of it was saved, frozen, and washed using pretty standard methods using sieves. And the hard parts were enumerated and identified to the lowest taxonomic level that was possible using reference guides. And from these information, we calculated a series of metrics, which are pretty standard for these diet studies. We have frequency of occurrence, which is a presence-absence metric. We used a prey-specific number, so this accounts for the number of prey that were consumed or estimated to be consumed in the number of scats that they appeared in. We also calculated a simple species richness, so just the number of species that showed up within a particular sample. And we estimated the minimum number of individuals that were consumed within that sample as well. For the DNA metabarcoding, we first had to figure out if it came from a gray seal or not, because gray seals and harbor seals are sympatric in the New England region, and they can co-occur during a lot of the period that we were sampling. So we used an assay with cytochrome B, which is able to distinguish between some of the seals, and it worked for us. So we used that to determine that 117 of the samples we collected initially were from gray seals. And these mostly reflected summer and fall diet, so keep that in mind as we're moving forward. Once we had these gray seal samples, we then amplified a small portion of the cytochrome oxidase 1 gene, which is pretty standard for metabarcoding studies. And we developed these primers for fishes, invertebrates, and birds using reference library for prey that are well known to occur in that region, as well as from anecdotal accounts from some of my collaborators. We selected a sample of 99 of these for DNA sequencing that were sequenced by the University of Rhode Island. And we used KIME2 for the bioinformatic processing. So basically, we took the raw sequence data, cleaned them up, combined the pairs of the reads that are in the forward and reverse directions, and then identified those quality-controlled reads against a curated reference library. And from the cleaned-up data, we calculated, again, a presence-absence metric, this frequency of occurrence. Again, because you can't directly quantify the prey data that are obtained. We instead used a relative read abundance metric to get an idea about proportions of prey that are showing up in the DNA data. And again, we calculated species richness as just the number of different species that occurred in the samples. Now, for stable isotope analysis, it was a little bit more involved because there weren't that many people that had done fecal stable isotope analysis at the point that I was doing my dissertation research. So one of the first things we had to do was see if fecal samples needed to be lipid corrected. For anyone who's worked with stable isotopes, it's pretty standard to correct for lipids because they can kind of downweight your carbon isotope values, which will impact kind of the results that you get and the inferences that you can make. So we used a subsample of our fecal samples that were confirmed as originating from gray seals and experimentally tested them with a chloroform methanol solution in a sonicator to see if they needed to be cleaned. And we ultimately found out that the change in the carbon isotope values between the experimentally cleaned samples and predicted from mathematics was not significantly different. So we were able to just mathematically correct the remaining samples that we analyzed. We also wanted to sieve the samples before we analyze them for stable isotope values. The Cape Cod beaches tend to be pretty sandy, and it has a very similar texture to the actual seal scat. So we wanted to make sure that we were analyzing organic matter as opposed to kind of the sediments that are in the similar area. So we retained the smallest fraction as we assumed that was where the organic matter was, and testing did confirm that. And then we did a pretty standard analysis on an elemental analyzer system and obtained the carbon and nitrogen stable isotope values relative to air and PD-belemnite standards. And for the mixing models, we also analyzed prey muscle from 17 species. This was informed by a previous effort that I was involved in that were collected from trawl surveys off the coast of New England. And of these, we used 10 as sources in the mixing model, and we used clustering methods to reduce this 10 prey species into 4 functional groups, just so that the math in the mixing model was a little bit more stable. And another thing that you have to consider when you're working with stable isotope mixing models are trophic discrimination factors. This is a mathematical correction that you use to account for the biochemical conversions that go on when you go from the prey tissue to the predator tissue. So we tested 4 different potential trophic discrimination factors, or TDFs, to use for the study because no one had yet determined a trophic discrimination factor for seal poops. These TDFs tend to be species and tissue specific, so it's kind of important to get as close as you can if you aren't able to do that yourself. So we tested 4 different factors. We used coyote, snow leopard, tiger, and then kind of a standard environmental TDF, for lack of a better term. And we found, based on the distribution of points in the system, that the coyote trophic discrimination factor was the most appropriate for our samples relative to the sources. So we used that going forward. And we ran 3 different versions of the mixing model in the program Mix-SciR. So again, we had 4 prey sources that were basically 4 different functional groups. We had salmon, squid, and mackerel, which are known prey types for gray seals. And we had a combined demersal fishes, which included flatfish and agatids and skates, since these are also well-known prey species that are consumed by gray seals. And they're also pretty isotopically similar, so you're able to collapse them and again, not overfit the model. And again, we wanted to see if providing information to the model kind of in the prior stage will improve the inferences of the model at the end. So 2 versions of the model were informed by either the hard parts data through the prey-specific number and one by the DNA relative read abundance data. The first model had uninformed prior, so it considered equal abundance to each of the prey sources as opposed to kind of this informed information from the other 2 methods. Finally, we looked at fisheries data and overlap. So as I previously mentioned, we got commercial fishing data for Massachusetts from an automated program through the National Marine Fisheries Service. And we obtained data for the previous 5 years to sampling to get an indication about kind of an average trend as opposed to just the specific year we were working in. And we computed these averages for both landings and value in US dollars. And to get at the potential overlap between seals and fisheries, we calculated this Morrissey to Horn index, which estimates the proportion of a prey that are consumed by the predators, in this case, seals and fisheries. And it gets at this index that varies from 0 to 1, with 0 meaning no overlap, 1 meaning total overlap. And it's often considered that I think a value over 0.6 is considered substantial overlap. And we calculated this overlap index with different metrics. We used the frequency of occurrence and the abundance metrics that we had calculated previously. So, moving into some of the results, with the hard parts data, we found low species richness in the diet. Nearly a third of the samples that we ended up analyzing were empty, so they had no prey remains in them whatsoever, which is kind of unusual. And for those samples that did have prey in them, we only found a single species, which again, for a marine mammal diet study is a little bit unusual. And when you look at the composition of prey that showed up in the samples, it's overwhelmingly attributed to sand lance. So this is a compositional bar plot where each of the colors represents a different taxa that's consumed by the gray seals. And so you'll see here these gray boxes are for sand lance. And you'll notice here if you read the taxon labels that a lot of these are not to the species level. That's because a lot of the prey remains were either broken or eroded because of digestion, and so you weren't able to confidently assign it to a specific species, which is a little unfortunate when you're trying to provide this information to managers. So again, a lot of this was attributed to sand lance. Some other common prey that we found included gadids, which could have been cod or hakes, we don't really know yet. And skate was another important prey type here. Moving on to the DNA metabarcoding results, we did find a higher species richness. So from the same set of samples, we found that instead of less than 1 species per sample, it was more like 7 taxa per sample, which is a little bit more what you would expect when analyzing pinniped diets. Instead of the about 7 prey that we identified previously, we found 21 different taxa, including several species that had not been previously recorded as prey of gray seals in this region before. But when you look at the relative reed abundance, again on these compositional bar charts, it's overwhelmingly attributed to sand lance. In fact, over 94%, I think it was, of the reeds that we got were assigned to sand lance, one of the 2 species that are common in this region. And a lot of these taxa showed up in 1 or 2 samples with very low reed abundance relative to sand lance. When we look at the stable isotope values, again, no one had really done this kind of work before, so we first reported the kind of values that you get. So we found kind of an average stable isotope values for seal scat of -19 for your carbon and 12 for your nitrogen. So at least for the trophic level information, it's pretty consistent for a mesopredator or a top predator, depending on where you're working. And we also had the stable isotope values for the prey sources plotted here as the crosses with the error bars in both directions. And it was reassuring that these values were pretty consistent with past stable isotope studies in the Gulf of Maine, kind of Cape Cod region. And you'll see that there's a fair bit of overlap in the biplot. Each of the black dots here is an individual seal sample, and these are the prey sources with their errors. We distinguished demersal fishes because they had higher nitrogen stable isotope values, which is what you would expect for predatory fishes. And sand lance and squids were denoted by lower nitrogen values and more negative carbon isotope values. So this kind of helped us with these stable isotope mixing models, or so we thought. All 3 models estimated that sand lances and squids were the most frequently consumed prey. So you'll see here on this density plot 4 different taxa in these 4 different colors, but overwhelmingly, over half of the estimated contribution can be attributed to squid, and almost as much to sand lance, with minimal contributions estimated from demersal fishes and from mackerel, which is something that you would expect because we didn't really see a lot of mackerel. And given the overwhelming abundance of sand lance through the other methods, we're not too surprised that there were lower contributions of demersal fishes to their diet. But it is important to note here that the confidence intervals that were produced by the modeling program did tend to overlap. So it suggests that because of the high isotopic similarity between the prey sources, that there's a little bit less confidence to say, what's actually going on here. The one thing that did stand out isotopically were the mackerel, but because they were so minimally consumed, it didn't really alter the model results too much. So when we take all of the information that we integrated together and compared it to commercial landings data, we're going to walk through this table because it is a lot of numbers. So we'll focus on sand lance first. As I hope you've gathered by now, they were pretty important to gray seal diet, showing up in pretty much every hard parts sample and every DNA sample, and were pretty, estimated to have a high contribution in the mixing models. But in terms of fisheries data, sand lance are not really commercially fished, and in fact were not fished after 2014 commercially in Massachusetts. So of course they would have a lower catch and value associated with them. Now the higher value fishes, such as the demersal fishes, had much lower contributions estimated by all three of the methods that we use to analyze seal diet. And if this kind of held true for squids as well, where there's minimal contribution estimated to the diet, except with the stable isotope mixing model, but also kind of they represented a lower proportion of the catch and value for the fisheries in Massachusetts. And when we calculated these Morosito Horn indices, we found that they estimated overlap on the order of less than 0.01, which suggests very minimal overlap between seal predation and commercial fisheries effort in Massachusetts, which is pretty reassuring. So when we consider everything that we learned from this study so far, we found that complementary approaches provided an updated picture of seal diet and a largely congruent picture of seal diet. So all three methods estimated that sand lance was an important prey resource to gray seals during the summer and fall months. As you would expect, using different approaches, other prey taxa were considered important with kind of method-specific differences. With the DNA, we found that flounders as well as sea robin were considered important prey to gray seals. With the hard parts, we found skate, and the squid was estimated as an important prey resource through the stable isotope analysis. And again, we found limited overlap between seal diets and commercial fisheries during this study period. So again, focusing on the summer and fall months, which is pretty reassuring because it was long held that seals were potentially over-consuming some of the prey species that fisheries tend to target in this area. And when we take a step back and consider my work in comparison to other recent studies of seal foraging ecology and diet in this region, we again see that there's a pretty high level of agreement. So a previous effort to use mixing models started with kind of a similar framework, except they didn't include mackerel as a prey resource. We ended up adding it in because mackerel occupied kind of a unique part of isotopic space that wasn't previously considered. So it did kind of overfit the model, but it didn't really alter the results since mackerel wasn't a large contributor to any of the diets. And the other major difference here is that we used SCAT stable isotope values as opposed to Lanugo, which is the prenatal coat for pups. And we did find that our DNA barcoding results were also pretty much in agreement with other DNA barcoding efforts that use different marker types. So these studies with fishes and invertebrates also supported kind of a similar diet for gray seals, where they're eating benthic prey, schooling prey, and not really anything that's commercially important, which is, again, pretty reassuring for the information we want to provide to managers. And if we consider what we learned from each method, hard parts is pretty reliable, again, because you can estimate the number consumed and the biomass that's consumed, but it did, in this case especially, only allow us to identify a small number of taxa, which is why you then use other methods to complement this. So with our DNA barcoding results, we did find a higher species richness, which is one of the things that you would expect. We also clarified quite a few of the species identification. If you remember from our previous bar plot, flatfishes weren't really identified past the family level. We were able to identify a couple specific species there. And we did, again, identify several new taxa, which hadn't previously been recorded before. And again, the mixing models emphasize squid and sand lance as important sources to seal diet. And this isn't unsurprising, given that sand lance is a major contributor to diet for a lot of carnivores in the Gulf of Maine ecosystem and indeed across the range. But again, I'll note that the prior information that we provided, the mixing models, didn't actually help to resolve the estimated contributions too much, again, because the prey were very isotopically similar. When you plot out each of the individual prey groups, for example, for the demersal fishes, they did tend to overlap quite a bit. So maybe there's other taxa that could help to resolve this later, or maybe mixing models aren't kind of the be-all, end-all tool to use in this particular system. So some other methodological considerations here. I will point out that we did use a single tissue type for all of these analyses. They were all based on seal scat, which isn't always the case for people that tend to use integration of different methods. You typically use the prey remains from a scat or a stomach sample, and you pair them with stable isotope analysis, for example, of muscle tissue or something with a longer turnover rate. That way you get the short-term information about what exactly they were consuming, as well as getting kind of a broader scale trend over a longer time frame. And so here, what could potentially help to solve the kind of short time frame that was reflected by all three of our methods is to include information from either stabilized setup analysis of different tissue types or with fatty acid analysis. And some of my colleagues at NOAA's Northeast Fisheries Science Center are investigating fatty acid analysis to kind of complement this work that I had done previously. And I'll note here that the DNA metabarcoding did improve our species richness, but some of the IDs that we got were a little suspicious. So I'll point out here two genus level identifications, this Epinephrylus and Pristipomoides. These are more warm water taxa. So they could have been vagrants that were brought up by the Gulf Stream and were kind of left to kind of wandering Cape Cod. They could also be just kind of errant identifications based on the reference library that we had. We used a curated reference library called Midori for the metazoan animals in our study. So it has animals from across the globe. So it's not 100% perfect, but it's a lot better and less biased than constructing your own reference library, which is what I did during my master's work. So it could be that these are, suspect identifications. They could be misidentifications for related taxa in different genera or different families that are actually present in the Gulf of Maine ecosystem. We aren't really sure yet, but it does kind of bring to mind that some taxa were expected based off of observational work from my colleagues, but we didn't actually get DNA sequences from them. And this included black sea bass, herring, and ocean pout. So some of these weird little identifications may be associated with them, or they could have just been sequencing artifacts that were misidentified. We aren't really sure just yet, but it's something to consider going forward. And again, we found that seal diet was primarily composed of species with little to no commercial importance, which is, again, very reassuring considering that seals were hunted because it was thought they were eating very important ground fishes. As I've alluded to several times, ground fishes were a critical part of the Cape Cod financial system. But if you look at fisheries landings now, a lot of it's based on invertebrates, lobster, sea scallop, different types of scallops and mussels and whatnot. These are kind of the major financial drivers for fisheries in the area, but that shouldn't discredit the fishers who are continuing to fish ground fishes and flat fishes and whatnot. So it's just something to consider going forward that it may not currently have a lot of overlap, but it is something to consider going forward, especially when we consider that seals tend to be eating prey that's no bigger than their mouth, usually. And so while they may be minor diet components, they may be eating a kind of critical prey class. They're eating juvenile individuals or smaller individuals. And if they're eating a lot of them, this could impact recruitment of those fishes going forward, which could impact the community that's available to not just them going forward, but also to the fishers that use those same resources. So it is something to consider. Now, we generated all this data, which is great because we updated our understanding about seal diet. But what's kind of the next step here? As I've mentioned throughout this talk, this up-to-date information is critical for managers, but they need to be able to take the well-informed science and translate that into policy decisions. But they can't make those decisions in isolation. They should be able to get that kind of information from other stakeholders as well. And when you work with politically charged issues, it can make coming to kind of a consensus agreement that's for the benefit of most, if not all parties, a little difficult to do. And if we consider seals, it's it's a little complicated. But recent sociological work looking at surveys in Nantucket, which is kind of a historical fishing town and still very active in fishing in the Cape Cod region, has revealed that attitudes among the general public, including fishers, is generally favorable, but there's diverging thoughts on potential solutions to how to deal with the increasing number of seals in Cape Cod, along with the increasing human population. So one organization that's working to kind of move towards an effective solution together is NASRC, the Northwest Atlantic Seal Research Consortium, which I've been involved in as part of my dissertation and afterwards as part of my postdoc a little bit. So this group was born from a series of workshops between scientists, resource managers, fishers, and other interested stakeholders, such as stranding network personnel, academics who study other aspects of seal life history and ecology. And together, they're trying to bring together different perspectives and ideas so that they can work towards kind of this better informed management of seals in this ecosystem. Gray seals and other seals in the US are still protected under federal law, so it's not like you can just go and shoot them. That is pretty illegal. So, you need to learn to live together, ideally, and that's what this group is trying to do. And they focus on research, education, and citizen science. So I wanted to end here by highlighting some work that I've done with NASERC to kind of help inform the management going forward. So we used similar DNA and hard parts methods to look at, and in collaboration with fishermen, to investigate aspects of seal bycatch. So young of the year gray seals, so these are individuals that have been weaned and are kind of on their first trip to sea, are often bycaught in different fisheries, especially gillnet fisheries in the New England region. And it's a little bit troubling because this could impact the persistence of the seal population going forward, but it's also bad for the fishermen because if they catch too many seals, their fishery will be shut down, which will have, of course, negative economic implications going forward. So we analyzed prey, again, using the DNA barcoding and the hard parts from the stomachs and scats of bycaught individuals to get an idea if they're actually consuming prey or if they're just being caught accidentally here. The different bar charts here present information from some bycaught individuals that were analyzed as part of this effort. And thankfully, they are eating not the target fishery, but they are eating some. So it's kind of an individual specific issue and something worth considering going forward as maybe a management thing to focus on. So to wrap up here, understanding the ecosystem role of gray seals was going to require additional research. It's kind of weird to think that marine mammal work still has kind of legwork to go. So we helped set the foundation here with this work and some other studies that I've been involved with, but it would be useful to establish a long-term monitoring of seal foraging habits in the New England region. And this should include harbor seals, which are actually decreasing in population size in New England, and there's not really a clear reason why, but it has been suggested that maybe competition between gray seals and harbor seals may be part of this, but we don't know, just because the information isn't there. Additionally, it would be useful to examine seal and shark interactions and competition. As I mentioned at the beginning of the talk, the increased presence of seals is thought to have increased the presence of sharks in the region, but also other species of shark act as competitors for some of the same prey resources. Spiny dogfish is really prevalent in the Gulf of Maine region, and they act as kind of on a similar trophic level to gray seals, and gray seals sometimes eat those spiny dogfish. So there's kind of weird ecological interactions that are going on, but we don't really know what's happening. So it's worth investigating. I think another great step forward would be to focus on energetics of gray seals. We have diet information, but we don't really have great consumption estimates just yet for the species. So this kind of work I think would be valuable going forward to better inform ecosystem models that are used by managers to kind of manage consumption and fisheries information. And again, all this informed science that we're providing will help to improve conservation and management of the species and the ecosystem that they live in. So I just want to thank all the many people that were involved in making this effort possible, and I'll take any questions. So thank you. Yes.
I'm just trying to figure out if you think squid are an important part of the diet of gray seals. So it seemed like your hard part data was like 4% frequency of occurrence. Your stable isotope was like, oh, it's like half their diet. So then if any of that's true, somewhere in between, why was the read depth so low in your metabarcoding?
Yeah, that's pretty good. It was a little complicated. As I kind of alluded to, squid beaks can be retained in the gut, so they get trapped there, which is maybe why they had lower frequency of occurrence in the actual scat. At least with sea lions, when they eat squid beaks, they tend to be regurgitated instead of defecated. So it does produce that bias there. So that's one reason. It could be that the primers that we developed for the invertebrates wasn't really great at capturing squid DNA sequences, and so you would have lower read depths for those species. We don't really know, but we did make sure to include the different species of squid. There's the Doryteuthis and Ilex squids that are in that region. So they were there to inform the primer development, but the actual sequencing, they didn't show up as much. But then I think maybe the stable isotope results could have been, again, because of that isotopic similarity between sandlets and squid. They were pretty similar carbon-wise and not that different nitrogen-wise. So that whole isotopic similarity between all those prey sources kind of leads to kind of those fuzzy results and the kind of diverging results, as you pointed out, between the stable isotope and the hard parts and DNA data.
Right. And they're not benthic. I mean, would a gray seal be good at catching squid?
They certainly try.
Thank you.
Yeah, I think the answer is yes, they're good at catching squid. I think people tend to focus on the fishes that they're consuming.
I see. Thank you.
Can I
Yes.
follow on that? I had to write my questions on here. Following on that, I was looking at your isotope plots. I mean, do you think that compound-specific isotope analysis could discriminate squid from, sorry, what was the one that they were similar to?
Salmon.
Yeah, sandlands, a little bit better? Like, do they have different amino acid contributions to a diet where that
That's
would work?
a great question. Other people in my lab at LSU did do compound-specific work in different systems, so it's possible that compound-specific isotope analysis, for those who don't know, analyzes stable isotopes for different amino acids, could tell those prey sources apart. Yes. But it could be something worth investigating because maybe that will have that advantage over bulk stable isotope analysis to tell those prey types apart. So yeah, it could be considered going forward.
Okay, sorry, I get excited about this. I tried some of this stuff, and my collaborator did mixing models, and I wasn't quite up to speed on how it all happened. So I had maybe a methodological question, which is just two, actually. Your trophic discrimination factors, you surveyed 4 and you chose coyote. What makes that decision happen? Like, what is the determinant that tells you that this is the discrimination factor that matches most closely?
Yeah, so you basically want to look at the distribution of your points in the space, kind of that isotopic space. If you remember back to the little multicolored plots here, you want to make sure that your predator values do not fall outside kind of that confidence area, which if you, especially with the environmental TDFs from posts, they did fall outside that area, so that was a pretty poor fit. So yeah, you want to just make sure that everything is kind of in the same isotopic space. If things get adjusted too much one way or the other, it will negatively impact the results. And so yeah, we were fortunate enough to find canid fecal TDFs, which is what those coyote ones were. The ones that we tested for snow leopard and tiger were also fecal isotope values, but those were animals that were in captivity and they were fed kind of a pretty standard diet, whereas those coyote values were from animals that were roadkill, so they were kind of more wild. So maybe there's the taxonomic similarity there, the canids and fosid seal, that made it a little bit better fit. But it could be something worth considering for your system in particular, to find kind of the best taxonomic fit and the best tissue fit for the system that you're working in.
Interesting. Okay. Cool.
I was just going to ask about, how did the carbon emission values shift in the prey in that digestion process? Right? I mean, they weren't incorporated into the seal, so they're not shifting because of that process. So is it just the process of breaking down the prey? Somehow the aspect values are shifting in particular directions? Or?
There's, that fractionation that occurs when you consume an animal. That's, I think, what those TDFs are trying to account for most, is that step, the 2 to 3 parts per thousand in nitrogen that's estimated, and the 0 to 1 in carbon isotope values. So that tries to account for that.
But in this case, it's just in the poop, right? I mean, it hasn't actually been incorporated into the tissues of the seal predator, right? So you're just looking at the prey that passed through the predator's digestive tract, right?
Mm-hmm.
So the remains that come out. So, I mean, I was just curious myself, like, how is that process causing the carbon and nitrogen values to change from the prey itself before it got digested?
Ooh, that is a great question, and one I don't really think I know the answer to right now. Because there's, it's an interesting system to try to work with when there's not a lot of information there. That's something to consider going forward, is like what exactly is happening. Yeah. Yes.
Has this been done on other species that you can compare your work to, or is this kind of the first, at least fosid?
Yeah, so some of the studies that I referenced earlier that integrated multiple different methods to look at diet, I don't remember if they did fecal isotopes. I think they used muscle or some other tissue to, again, get that longer time frame to kind of complement the short time frames. So it may not be directly comparable just yet, but I think it helps set a framework going forward for people to kind of integrate either 2 or 3 methods together to get kind of the best picture going forward. And folks that have been doing that with other species, like I read a study recently with, that did this with cookie cutter sharks, and they also produced pretty similar results using information from multiple techniques. So I think the framework is more important than the actual direct comparison, one-to-one. Yes?
Do you have any guesses why there were so many scat samples that didn't have prey items? Is it because of the capture method, or?
Yeah, so the scats themselves were deposited on the beach. We tried not to disturb the seals to get the samples that we used. Samples can just be empty because they're empty. They may have passed any prey remains in a previous event, or they aren't passed yet, so they're still in the digestive tract. Could be depending, again, on what they're eating. If they're eating a bunch of squid and it's stuck in the upper GI, it could be regurgitated again instead of defecated. There's kind of a bunch of different reasons why there could be no prey remains that showed up. It was just a little jarring to me to go from sea lion samples, which had, like, on average, 5 or 6 or more different prey types in them, to see these gray seal samples that had nothing in them. It was just unexpected for sure, but that's what happened. Yes. Thank you. Oh, that's a great question. Yeah, that's actually one of the big missing pieces of information. There's not been a lot of dive records for gray seals in kind of New England waters. There's work that's been done in Canada and the UK, but less so in the US waters. So we don't really know what they're doing with regards to that. In terms of prey switching, it's a little hard to know because a lot of the studies are, I guess, more contemporary, if you can consider 1995 contemporary to 2010, 2020. So we don't really know what they were doing prior to the 1990s. That information may exist and it's buried in some historical records, but I didn't find those studies if they exist. So we don't really know what was being done before kind of that initial report in 1995. We could assume that they were eating pretty similar prey items, which is what we would hope to see. So again, a diet of largely benthic prey, schooling prey, cods, sand lances, hakes, all that stuff. But again, because we don't have those historical records like we do with California sea lions on this coast, we can't really say, oh, there's been a major shift in their diet from this prey source to this one, or something commercial to non-commercial to make up for the lack of things. We don't really know that information, which is why we're kind of setting the baseline again instead of building on kind of historical records. Yes?
A cool idea, and we think too, your sampling, you sampled scat across quite a few seasons, I think it was. So did you see seasonal changes?
Well, except remember that when we did the cytochrome B assay, we found that most of the gray seal samples were concentrated in summer and fall. So we weren't able to really say that there were differences between summer and fall diet versus winter and spring. Some of the other studies that were done, one of the DNA metabarcoding studies, they did have winter samples, and those are a little bit different, but they also sampled at one of the breeding colonies, which is close geographically, but is in a slightly different biogeographic region. So there's, of course, different prey types that you would expect in those kind of conditions. So there's, all these kind of confounding factors that we can't really tease apart just yet, but that's why kind of continuing this sampling with multiple techniques is going to, again, kind of, maybe help us tease apart what's actually going on versus what's kind of confounding one another. And there's no questions in the Zoom chat for anyone. Awesome. Thank you all. Oh, fun.